Well, good morning, everyone, and thank you for joining this webinar. We've labeled this "Thinking Outside the VAT," so the insulin VAT or the box, and really to think about new approaches to manufacturing insulin, and how this could be an opportunity for improving and addressing some of the challenges that many of you are familiar with when we look at access to this life-saving medicine globally. So my name is David. I'm an assistant professor at the Division of Tropical and Humanitarian Medicine at the University of Geneva and Geneva University Hospitals. And today's webinar is part of a project that's supported by the Swiss Agency for Development and Cooperation, looking at raising knowledge and awareness around some of the issues around access to insulin and other medicines for non-communicable diseases. This webinar will be recorded, and we will share the recording with you because we have two speakers from one speaker from Australia and one speaker from New Zealand. We had challenging issues finding a time that was best for everyone. So for our colleagues in the US and in some colleagues in other areas of the world, we said that we would record this webinar and then share the recording. So once the recording is done, we'll share it with all of you, and please feel free to share it widely. So just to give you a bit of background, so the Division of Tropical and Humanitarian Medicine is based at the University of Geneva and Geneva University Hospitals, and has as its aim to develop partnerships, both in terms of clinical work, projects, and research with a variety of colleagues throughout the world. And the area of focus in terms of research is on access to medicines with two disease categories. One is neglected tropical diseases, and the other is non-communicable diseases with a specific focus on diabetes. On the panel today, we have Molly Lepesca, who, together with Margu and myself, lead the Addressing the Challenge and Constraints of Insulin Sources and Supply study, which is a study that's been ongoing for many years now, that has tried to address a variety of issues with regards to the issue of access to insulin and highlight some of these issues. As I mentioned in my introduction, this webinar today and hopefully future events are part of a new initiative that we've just launched with support from the Swiss Agency for Development and Cooperation, with this idea of having an NCD policy lab to highlight certain key issues and innovations in the area of access to medicines. And this project, Improving Access to Medicines and Technologies for Non-Communicable Diseases, will use diabetes as a tracer condition. And so this webinar today is one example of what we're going to try to do within this NCD policy lab to highlight challenges, to highlight possible innovations and solutions, and to share these with a wide audience. So if we think about issues around access to insulin, we have a wide range of challenges. So if we look at research and development and innovation, we see that innovation has always resulted in higher prices for countries, for people with diabetes. And do we have innovation that's adapted and affordable for low and middle income countries? And we could also have a discussion, are we seeing real innovation or just marginal innovations? The topic for today, manufacturing. As many of you know, manufacturing of insulin is dominated by three multinationals on the global market. And also with insulin, we face this complexity of producing a biological product. The next stage, marketing registration, regulatory processes for biologicals are more complex, and we also lack capacity in many contexts to appropriately assess these products. And then again, a topic that many of us are familiar with, the selection, pricing, and reimbursement of insulin, with a lack of government policies on appropriate selection, many countries not having appropriate pricing mechanisms, and is insulin reimbursed and made affordable to populations. We also face a challenge once the insulin arrives in the country to supply this insulin throughout the country, as well as earlier on challenges within procurement processes and markups within the supply chain. The last three areas really focus on the health system, and again, diabetes care in many contexts is challenged because of the weaknesses of health systems. So where is diabetes care provided? Is insulin available throughout the system, throughout the country? Are healthcare workers appropriately trained? Where is insulin available within the country and health system? Is it only in large cities? Is it only in hospitals? And then finally, the most important stage of all is when the insulin is used by an individual in their household, in their daily lives. And is this insulin available, affordable, and can people access the care that they need? So really, what's interesting and the reason that this webinar came about was that in thinking about this pathway of insulin, we've sort of been stuck with the fact that for manufacturing of insulin, we have this current model of 3 large multinationals, a few biosimilar companies, and other areas within this pathway have been discussed. Different issues have been raised both in academic literature discussions, but very little has been focused on this issue of manufacturing, and so that's why this webinar came about. And really, the aim of today is let's think outside the box and see, can these innovations that we'll have presented to us very shortly by 3 experts, and then the perspectives and insights from other experts, is this something worth pursuing? So really, the aim of today's webinar is to present innovative approaches to making insulin, and we have 3 interesting speakers, and I'm very grateful for their participation and the insight they've brought. So we have Ilya Vensky, who's the CEO and founder of Helico Bio. We have Xavier Frépas, who's a clinical consultant at Biosourcing, and Alex Kelly, the founding director of Neosyn Bio. This presentation, this webinar, is by no means an endorsement of the companies or approaches presented, and really the aim is to have an academic exchange on this. So after the presentations by our 3 colleagues, you'll have a chance to ask questions in the chat. Then we'll have perspectives and insights from 3 leading experts in terms of different issues around access to medicines, and I'm extremely grateful for their time. So we'll have Julien Potet, who's a senior advisor from MSF's ACCISS campaign, Molly Lepesca, who's a project manager for the ACCISS study based at Health Action International, and finally, Professor Amanda Adler, who's a professor of diabetic medicine and health policy at the Radcliffe Department of Medicine at the University of Oxford. And then finally, we'll have a few responses from the perspectives from Alex, Xavier, and Ilya, and then some closing remarks. So I'd like to thank you for your participation, and very much looking forward to the discussion. And I will stop sharing my slides and hand over to Ilya.
Good. Excellent. Am I in?
You're in, and we see your slides.
Okay, wonderful. Well, thank you for the opportunity to present Helico. This is very exciting for us. So our top-line vision, we use plants as our production methods to produce existing and novel biologic compounds. So that's really kind of a top line of our approach. In a commercial sense, core capabilities value proposition is a low cost, high margin production or manufacturing at small volumes. And by small, I mean, not sort of compared to, Novo Nordisk, but let's say kind of a biotech volumes. And our 3 lead indications right now, it's analog insulins, universal antivenom, and specific cancer treatments that I'll explain in a minute. Just a little bit about plant-based science. So we use Nicotiana bentaminiana, which is a kind of distant relevant of tobacco plant. We use it the same way industry uses lab rats or mice, and this is a kind of a model organism for plant science for the last 50 years. Not going to go into the problems. I would rather cover solutions, but basically the problem that we think we're addressing is cross-border transactions, so setting up local manufacturing using highly scalable plant system. cost of local biosimilar setups, factories, production facilities are still very expensive to set up from a capex point of view, and the cold chain. So poor cold chain is a really serious issue. So our solution, growing plants in confined environments, is cost efficient, modular. So we exploring even doing it in shipping containers, but for sure in kind of modular environment. It's controllable. So basically you have semi-automated systems that control the production, and it's transportable. A little bit about our science and background of science. So we have a proprietary system to use sequence optimization before we start working with our plant. We use introns to modify the plants. Then when we have our genes, we insert it in the plant tissue. This is pretty standard transit expression systems. But our system kind of allows for quite significant yield improvement, and the plant system being known for low yields. And this is really what we're kind of addressing with our specific technology. And our modular workflow, which we feed in back all our experiments, is currently in US patent examination stage. We also managed to modify our plant, basically our host organism. We used bent or Nicotiana benthamiana, but we created our own strain. It has extremely high biomass because it doesn't have a stem. It's actually also drought resistant, and because of the size of the plant, you can put 12 shelves instead of typically 3 shelves. And that's dramatically increased biomass and productivity. And we're also putting a novel plant variety on our plant later on this year. And we're also experimenting right now with supply chain efficiency. So again, as I said, we use shipping containers. So we use various innovations through partnership with other companies, specifically in solar power and hydro panels for water delivery. So I'm not going to go in details of the slides, but basically all of this innovation allows for extremely efficient timeline to look at cost of production and development of production line. So our goal is to basically come up with a system that will allow for GMP production within 6 months. From let's say analog insulin or a novel compound to actually being able to go into the GMP stage. So this is extremely fast compared to mammalian CHO lines or E. coli. So now to insulin. Our goal is obviously to develop global access to insulin. The industry has a tremendous size, but what interests us the most is that quite a few studies shows that approximately 15 million patients don't have access to insulin or have limited access to insulin, in majority of them in low and middle income countries. We really believe that we're kind of leading a major commercial breakthrough since the development of recombinant insulin in E. coli. Plant's incredibly efficient, and adding all the innovation that we're working on, it would be very unique new system. But plant-based insulin is not new. It's basically technology exists for 20 years. It's never been commercialized for a variety of reasons, mostly because it was developed by academic research groups, and barrier to market was too high, and they didn't have tools and innovations that are available today that we use, specifically in computational modeling. And the biggest issue and barrier with plant-based insulin, and actually any type of expression in plants, as I mentioned, it was poor yield. So this is what we are extremely excited to provide to the current solutions. So the targets what we're looking at in insulin, we're looking at off-patent targets. Basically, we're starting with the Lispro, but we can quickly develop any other analogs. The science mostly known, so we just optimizing what is already being done in a pretty vast research. It will be expressed in a helical plant and will create highly simplified production method. The result will be high yield and lower production costs. We didn't want to share specific numbers because we're still working on it, but it looks incredibly competitive, and will be scalable and transferable, which is really the main point. And we can work on multiple analogs. And our commercial access to the market, as we see it, would be biosimilar pathway directly in the specific markets. And we already have a few conversations with health boards or tech transfer via an NGO. Just to show you where we are. This is our lab in New Zealand and soon in North America, and we're in the process of raising capital at the moment, and we're hoping to do a first scale-up production in 2025. We're also very excited. We were looking at after sort of investigation of a market need and how we can improve insulin target. And we decided to look for a novel insulin, especially with a better thermostability. So we have a partnership with very interesting company in Colorado. So we choose a specific thermostable target. Will begin expressed in the helical plant, but it will be novel indication. It's a single chain insulin. We already done all the R&D, very exciting work. I'll show you some results. And again, it will be scalable, transferable, and we will try to do directly to the market through health boards. Unlikely it will be done through the NGO as we kind of see it, because it's a novel indication. So we'll have to do clinical trials via partnerships in US, and also looking at some opportunities to start in a potentially animal health market. But understandably, thermostable insulin would be an amazing product for to solve the problems. So this is just a few data points from the target that we choose. Very high thermostability, extremely concentrated, so it's U1000. Very high bioavailability, and you can see kind of in this graph on the right, the blue line is basically straight up to the 800 hours, so 28 days and 40-degree temperature, and it didn't basically affect it. This is our first results. So you can see sort of, I'm not going to go kind of in details again, but you can see sort of on the column 5, this is actually expression in our plant around, a band around 10. This is kind of really strong indication of presence of insulin. It should be 5, but because it's a single chain, it's actually 10. So very exciting work, and we're really happy with results, and we'll start optimization now. So sort of like really what are benchmarks that we see is our delivery. So for biosimilar, the Pro would be market-ready products, low production cost, scalable and modular. The cons would be downstream is complex. Our idea is use conventional downstream processes. So we're investigating it right now, but it's a pretty complex. So we're still working on it, and regulatory costs are relatively high. For thermostable, basically the same pros plus solving cold chain issues, and the cons will be regulatory costs will be high because it's a novel indication. I also want to just quickly go through 2 other targets that we're working on. So we're working on antivenomous because it's partially in neglected tropical disease categories as well. So we have the same aspiration while we're working on it. It will be novel education. This is commercial work with our partner in US. And we're also developing a very interesting biosimilar of one of the cancer treatments that going off patent 2027, which is not even available in anywhere in the world besides United States and Europe. Quickly about team, Miguel Gomez Lim is our CSO, being in the field for 30 years. Colleen worked in CRO in Europe for many years, in United Kingdom and France. I have a mostly commercial background, and Xiaoming is now data scientist, science side, and that is all.
Thank you very much, Ilya, for that interesting presentation and this whole pathway that that Helico Bio has gone on. I think it's really exciting, and having had many exchanges with you over the past few years, it's great to see these advances. So we'll hand over now to Xavier Frépas, who's the clinical consultant for biosourcing. Xavier, the floor is yours. Your microphone isn't on, Xavier.
Okay, can you hear me now?
Yes, we can.
Okay, can you kindly, David, put my slides on the screen? Okay, so good morning or good evening, everyone, and thank you very much, David, for this opportunity to exchange and discuss what biosourcing is doing and how it could help making insulin more affordable for low and medium income countries. So as you know, for many years, genetically modified laboratory animals have been a very effective tool for studying the functional properties of genes, proteins, and other molecules. And their importance as human disease models in biological research cannot be overestimated. Such animals can be used in the study of pathogenesis and molecular feature of the disease, for identification or validation of new target, and for effective search and development of new drugs, including preclinical studies. At the same time, it is going to be the topic of this presentation, genetically modified animals are becoming increasingly attractive models in fields such like livestock farming, where genome changes can be used to correct economically important animal traits. Finally, and more importantly for us today, transgenic animals can serve as bioreactor for the synthesis of recombinant proteins secreted into milk, which enables production of recombinant proteins in substantially larger amounts and at much lower cost than the production of proteins in eukaryotic cell culture. Next slide. So this is a biosourcing team, a team of experts. They are really geneticists, highly talented geneticists and veterinary and vets. So a very, very, very science-oriented team. They are based in Liège, Belgium. And it's really an absolutely outstanding team, really great scientists. Next slide. So that's me. Just one word to say, and you can skip it, that I've been in the pharma industry for 40 years, and I've developed expertise in biosimilar, and that's the reason why biosourcing asked me to join their team and to support demonstration of biosimilarity, which for this kind of product, with this kind of production method, can be a challenge, and will certainly be a challenge. Next slide. So this has been already mentioned by the previous speaker. Both Western and emerging economies face major issues in terms of cost, capacity to produce biopharmaceuticals. And that has been said and will be repeated, I'm sure, by the other speakers. The capex for this kind of production using classical production method is extremely high and not something low and middle income countries can consider. Next slide. And as you know, also basically the biomanufacturing is basically concentrated in US, Canada, and Europe, with now China and India coming up. But if you see the share of countries or continents like Africa, it's very, very minimum at the moment. Next slide. So in the last decade, revolutionary changes have occurred in the field of genome modification due to the opportunity afforded by highly effective targeted genome editing and the simplification of this technology after the discovery of the CRISPR-Cas9 system. Next slide. So you can put, keep going. Yeah, well, here we go. Thank you very much. So just to keep it relatively short, this slide describes the sequence of events leading to the production of insulin and actually other proteins of interest. We have different steps. The donor cell fibroblast taken from ears of goats, and then the Cas9 system with the implementation, the insertion of the precise locus, then the clone screening for antibiotic selection. Antibiotic-based selection, as you know, uses an antibiotic to screen desired transformed cells. And this transformed cell will have a selected gene marker on the plasmid. Within that protein provides antibiotic resistance, allowing these cells to grow while untransformed cells do not grow. And the last element, important element, is nuclear transfer, a process by which the nucleus DNA of the somatic cell is transferred into a nucleated oocyte for the generation of new individual genetically identical to the somatic cell donor. And we have also some patented filtration purification process in place, so will enable, hopefully, to get a very high insulin and other proteins of high purity level. Next slide. So the thing is, it has been predicted a couple of years back that the use of mammary gland as a bioreactor for the production of recombinant proteins will be one of the most important areas of technology by the year 2020. We're there now, and the progress is evidenced not only by numerous experimental studies, but also by already commercially available drugs based on recombinant human proteins. Just two examples here on this slide. The recombinant human antithrombin 3 is produced from the milk of transgenic goats, the way we're going to use for insulin production. And the recombinant human C1 esterase inhibitor is obtained from rabbit milk. So when I first was introduced to this technology, I could barely believe it. I said, "Yeah, you're kidding me. You're not going to get drugs from the milk of transgenic goats." Well, yes, and actually not only this is done, but it's also validated by EMA and FDA. So there are drugs in the market. And I mentioned here drugs that are human medicine, but in veterinary medicine, there is another compound which cannot mention here that has been developed exactly using this goat milk technology. So this is not science fiction. This is here. It happens. And we are very confident that this technology will enable us to provide different types of protein, including, for the topic of today, insulin. Next slide. Well, we can skip. Next slide. So, as mentioned by the previous speaker, there are lots of advantages of moving away from the classical technology, CHO. We can produce large quantities, but we can adjust the quantity we want. That's another big advantage. The cost of goods is drastically reduced. So I think all the alternatives that will be discussed today, I believe, share the same pattern, that higher opportunities to produce big quantities and also low cost of goods. Next slide. So this is the thing. I worked in the big pharma and Boehringer, for instance, and when they build up a new facility, it was something like 800 million euros to build up a new facility in South Germany. So with this kind of technology, ours, and I guess the one described by the previous speaker, the level of initial investment is much, much lower. And that's certainly something important and lead to cost saving, significant cost savings. Next slide. So low cost of goods, that's very clear as well. That goes without saying. The low cost of goods being driven by low capex initially. That's the thing that makes it possible in many different countries of the world where they don't have the finances available to fund expensive drugs. Next slide. And this is the thing. We can decide to build a goat farm, a large one, a small one, in one country, in 10 countries. It's very easy, it's very adjustable and scalable. That's a big advantage. And that's something authorities in different African countries we are in discussion with are very sensitive to. And plus, also the element that they will produce themselves, so there will not be shortages of insulin, as recently countries like Morocco have experienced. Next slide. And the other element, we are in a green environment where we all have to save energy and be efficient and everything. These natural bioreactors are resource efficient. They don't need the level of energy consumption that the classical CHO plants need, and a significantly lower environmental impact, which may not be the most critical thing, but still as an important element to consider. Next slide. So we can skip this one. We have discussed it already. And just to mention that biosourcing has two priorities. On the left side, this topic of today's discussion is insulin, and this is a relatively advanced project. But biosourcing is also aiming at developing different biosimilars, and you have here the list of biosimilars that are being considered, being developed at the moment. Next slide. And we have already a lot of successful partners identified, and I'm not going to go through them, but we have the support of very excellent partners. So it is this big animal health company being a very important support to us. Next slide. And that will be it. If you need any more information, Bertrand Méraud, the Chief Executive Officer of the company, will be more than happy directly or through me to address any question or contact you want to have with him. And with that, I think I'm right on time, David, and I thank you very much for your time.
Thank you very much, Xavier, for that very interesting presentation. We'll now hand over to our last speaker in this first part of the webinar, Alex Kelly. Thanks, Alex, for being with us today.
No, thank you very much. That was both of these so fascinating. Hope you guys don't mind taking a technological step back a little bit as we reinvent the wheel. But I will share my screen now and talk a little bit about open science. Fantastic. Thank you. So I'm going to be talking to you today a little bit about open source protein manufacturing protocols and the open science movement in general. So open science is a global development to make the knowledge generated by scientific research available and accessible to all levels of society. So this sort of stems all the way from citizen science, anyone getting involved in scientific laboratories, to open research and ensuring that people have access to scientific journals. My experience in this is that I ran an open science lab called BioFoundry here in Sydney for 4 years, from 2017 to 2020. And we just provided a PC1, that's a physical containment one space that anyone can come and learn genetic engineering techniques for free. And we also ran projects seeking to synthesize proteins in an open source manner. So making all the things necessary for running a lab, things for making DNA, for sticking DNA together. But in that process, we came across some other interesting proteins that need to be made, most importantly, insulin. So we heard about the Open Insulin project while we were working there, and we thought that we'd love to get involved. And so I'll just talk a little bit about them and show a few slides that have been shared to me by Anthony DeFranco, one of their founders. Their basic institutional goal is that they're working to develop the first practical, small-scale, community-centered model for insulin production to make insulin accessible for everyone. They envision a world in which communities in need have local sources of safe, affordable, high-quality insulin, where people living with diabetes in their communities can own and govern the organizations that provide the medicine that they depend on to survive. So I'll talk a little bit about them, but feel free to look them up, the Open Insulin Foundation. They're an excellent group, and I highly recommend checking out their website. Their basic business model is they would gather community biolabs together and help develop open insulin protocols and production methodologies. And then they develop some some institutions to get to market that are a bit different from the current large-scale manufacturing. But I'm really here involved in the first two steps. And so the way that our side of the things look is we design vectors for making insulin. We engineer the microbes, we do the purification, we verify that it's working. And then some of our partners then move on to the manufacturing side of things. So when we got involved, we were just a very, very small lab with a whole bunch of secondhand equipment and not a lot of funding. And we ended up partnering with the University of Sydney to go to the iGEM competition, which is this fantastic international competition that gathers synthetic biologists from all around the world to develop methods and new technologies for manufacturing proteins in bacteria. And so universities and high school students from all over the world gather together and they present their ideas. And we knew that insulin manufacturing was by all means not a new technology. We've been doing it for 40 years. But the wonderful thing about iGEM is that everything that you publish online. It's free for everyone to access on their wiki. And we thought that it would be important to provide both the sequences for insulin on the iGEM wiki, as well as the methodologies required for making and purifying insulin. And thus the DISCO project was born. Or designing insulin to be single-chained and open source. So Ilya previously was talking about single-chain insulins, and we were also fascinated by these, both just as a way of exploring analogs in the market and the role that they play, which is in a lot of ways constraining the development of cheap insulins by continuously reinventing newer designer insulins. But at the same time, we sort of thought that there could be some benefits to this, and the cold chain transportation was one of those. So the main goals of our project were to generate awareness, simplify insulin production, and provide an open source solution. Now, I know that a lot of people in the audience know exactly what insulin and diabetes are, but for those of you who are coming from maybe a market side of things, I'll quickly just say that insulin is a protein produced by our pancreas that controls blood sugar levels. It tells our cells to take up glucose, and diabetes results from issues with insulin production or utilization. And you've seen how insulin is produced in plants and how it's produced in goat's milk, which are both fantastic technologies, but I'll take you all the way back to 1978 now, and I'll just quickly explain how insulin is manufactured in bacteria. First of all, we take the gene for insulin and some genetic machinery on either side. We put it in a circular piece of DNA called a plasmid. We then use antibiotic selection to select the bacteria that have taken up the plasmid, and then we grow it. We can grow it in a flask with just 100 mils of culture, or we could grow it in a giant bioreactor with 10,000 liters or 50,000 liters. We then take the bacteria out of the bioreactor, we break them open, and we get the insulin, which can then be purified by a number of different methodologies, the most common of which is called nickel NTA purification. We run the cell slurry through a resin, it captures all of the insulin on a magnetic bead, and we then wash off all the debris, and then we wash off the clean insulin. We can analyze it, and then we can dialyze it to get rid of all the chemicals used in the purification process and end up with our purified insulin. And so the basic structure of insulin is it's produced as one long chain called proinsulin made of 3 individual smaller chains. The C chain is cleaved off by the body and leaving the alpha and beta chain joined by these things called disulfide bonds. And this is our active insulin molecule. When we talk about single chain insulins, we're actually looking for a solution for cold chain transportation because these disulfide bonds don't hold the insulin together so well, and you heat it up for too long and it starts to fall apart. So a single chain insulin actually gets rid of the long C chain linker and joins the A and the B chain together. And so we ended up developing our own single chain insulin, which was not covered by any existing patents and provided the sequence online for free. We haven't done all the excellent validation steps that Ilya's done to show that it's structurally sound for a long period of time. But Ilya, if you want our sequences and want to do the same validation on it, it's free for you to use online from our iGEM wiki. We then explored 3 different expression platforms, from expressing it in the cytoplasm of E. coli, so like the big central chamber of the bacterial cell. The issue with that is that it forms these inclusion bodies and doesn't form the disulfide bonds correctly. We explored expressing it in the periplasmic space, which is a smaller compartment, but it does lead to the creation of successfully folded insulin, so you don't have to go through chemical refolding afterwards. And finally, we looked into beta subtilis expression, where you basically export the bacteria out of the cell and you can continuously harvest it in a continuous reactor, which technically should have a lower cost associated with the manufacturing. So I just want to thank everyone involved in the iGEM project, as well as my amazing team. And then at the end of 2017, we took that all the work that we'd done there and took it back to BioFoundry, which was still a very, very small closet. And I built up that lab over the next year, maintained the plasmids in our freezer. The following year, we designed new vectors for improved manufacturing, published our research online, and we were seeking out a FPLC to try and mimic industrial purification, to try and demonstrate from the start of a bacteria to the purified insulin how much it would cost. Unfortunately, COVID then ended up shutting us down because our lab, we needed about 5 or 6 scientists in a base the size of a closet. And so the plasmids are now kept in my personal freezer while I await a new lab space. So with BioFoundry closed down and no way to legally continue experiments in Australia, I decided to try and then publish everything that I'd learned over the course of the last few years on a new website. I started up a new company called NeoSynBio. So NeoSynBio's main aim is to eliminate the scarcity of medical supplies around the world by ensuring that public and private sector operators have access to the functional intellectual property they need to deliver safe, high quality, and cost-price medicines to patients. And I really focus on the genetic engineering side of things with this. So the basic goals of NeoSynBio is to provide all the knowledge necessary to cheaply start up your own biolab, including low-cost protocols, necessary equipment, cheap alternatives. And I provide the basics of synbio theory necessary to create bacterial expression constructs for biomedicines. So if you have a rare protein that you need to make and you live in a place in the world where you don't have access to it, I try and provide you with all of the information you need necessary to be able to produce it. Currently, we've written about 140 guides, and in the last year to date, I've had 17,000 visitors to the website from over 100 countries. Of course, some of those are probably from VPNs, but it's difficult to say. And the most popular page at the moment is G-block design, which shows you how to produce a genetic part for the expression of a certain protein. As for myself, I realized that my skills and knowledge were not sufficient to explain absolutely everything, and especially with the things that I'd learned in a low-cost lab. So I've gone back to university to work on a PhD now and improve the quality of the guides that I write. The wonderful thing about that is I have access to all this fantastic, expensive equipment that I can get trained on, things like the FPLCs that were far out of the budget of BioFoundry. My goal is to finish all the guides necessary for an insulin manufacturing startup, and then start up a new lab to continue to demonstrate and hopefully produce video guides for all of my protocols. So just coming back to insulin manufacturing and all the lessons that I've learned along the way about producing insulin in bacteria. The simple fact of the matter is that the synthesis side of things in bacteria is relatively easy. Making DNA sequences and getting expression stains is very cheap. However, the purification side of things is the thing which we run into a large amount of costs, especially around the purification. So getting an FPLC or getting large-scale FPLCs is very expensive. The purification resins that you use, like nickel NTA, is very expensive. You can regenerate them, but that takes time as well, and then requires its own GMP. And of course, getting GMP approval for your manufacturing plant is very difficult. And I can see why examples like doing things in plants or doing things in goat's milk could definitely get around some of those expenses. So in short, in bacteria, the synthesis is easy, but the purification is very hard. Thank you, everybody.
Thank you so much, Alex, for your presentation. And I should say that Alex was the first person who sparked, in a sense, this discussion and thinking around, should we be rethinking how we make insulin? So thanks, Alex, also for that. The floor is open for any questions, either from the audience or from our other panelists. I see that Damjan has a question. If you could just let me see. If you could just write your question, Damjan. Unfortunately, we can't open up the the mic for all questions. While we wait for Damjan to write his question, I don't know if any of our speakers have questions for their colleagues or anything else. I guess, well, while we wait for other questions, I'll ask the first question. I think, again, thank you to the three of you for your presentations and also for these exciting innovations. I guess you present a lot of the upstream elements to making insulin in different ways. We know that making the protein is the first step. We then need to get this in vials or pens, and then get this registered, go through regulatory processes, priced, prescribed, and then used. How do you see your roles as innovators in this space, addressing some of the downstream elements to ensure availability of insulin for some of the populations that most need it?
I can put my 2 cents, David. Downstream is hard. It's very hard, very challenging. I mean, we're addressing it with a few elements that known in plant systems. Basically, with a single chain insulin, it's the technology that we're licensing. It's kind of built in. So you know that's not really that challenging. With the conventional insulin, it's more challenging. So we're experimenting, adding a few linkers that might be degraded by cell itself. You know, that's kind of in a nutshell. The rest of it will be pretty much conventional ways. There is not really a lot of innovation in downstream and overall process besides open insulin project and a few other sources that we know is available. So I think cooperation between everyone on this online and broader would be essential.
If I may, David.
Yes, please.
Yeah, I think the reason why we picked up insulin as one of the first molecules we want to develop is, from a regulatory perspective, it should not be too challenging. With a lot of a lot of caution when I say that, because this is a well-characterized protein. It's not a monoclonal antibody. A monoclonal antibody, you can have all kinds of post-translational modification that can happen. And we're never quite sure this is exactly the same molecule as the reference product. For insulin, like pegfilgrastim, they are short molecules. They are well characterized. So we believe that the demonstration of biosimilarity from an analytical perspective should be relatively easy to make.
Thank you. We've had some questions and comments from our participants. The first question, I guess, is mainly focused on, for you, Ilya, is can you use mushrooms instead of plants? And then we've had a comment about the issue of financing for open science, and that this is the main struggle. So maybe that's targeted more for you, Alex. So I'll let you both answer those questions.
Ilya, do you want to go first?
Sure. Mushrooms are interesting for sure. But as I mentioned on the second slide, we use most studied and fully sequenced lab rat of plant science as our host, because that's simplifies a lot of methodology for computational side, gene design, and has abundant amount of scientific literature to proceed. We don't really know mushrooms that well, unfortunately, so that will be very challenging.
Thanks, Ilya.
And yes, in answer to the question about funding for open source, 100% correct. The thing we internally ran into was the fact that we really were not funded to any large degree, and so we had to be mostly self-supported. The Open Insulin project has now become the Open Insulin Foundation in America, and I believe, they now sort of have, I think, achieved what we in Australia would call charity status. So I think they're looking to try and work on crowdfunding and which is how they originally got their money, in ways to be able to reliably source funds for their projects. But that said, it is still a proven model that you can produce a biomedicine and make a profit, but not actually have intellectual property firewall protecting you from competition. Because you have companies like Biocon, Wockhardt in India producing large amounts of insulin, and I think becoming starting to creep up there in the market share that they produce insulin for, especially in India, and managing to at the same time, remain in business. And so whether it comes through public-private cooperation or newer business models that rely upon producing large amounts with a low margin rather than a small amount with a high margin, that might be the answer. And yeah, that is really my answer. I do have some questions as well for the other panelists, but I'll let you get to the other people to ask first.
So we have two more questions from our participants. The first one is, thanks for all the presentations. I am a person living with type 1 diabetes, and the first thing that came to my mind is that many people with diabetes have a sort of attachment commitment to whatever brand insulin they are prescribed by their doctor. Some can be very unwilling to switch from Humalog to Novolog, for example. Have you thought about how to get buy-in from insulin users themselves or from prescribing physicians? And then the second question, which is more focused on the market, is thank you for thought-provoking webinar. As noted, the insulin market is dominated by a few companies. How do we ensure that the new innovations create a more diversified market rather than enhancing production by the existing giants? So two challenging questions for our three.
David, I'm happy to address the first one.
Okay.
Because this is actually something we have seen experience with the old biosimilars. There is an initial reaction against biosimilars from patients, prescribers, and everybody. And then gradually, sometimes clinicians are forced to prescribe biosimilars by pharmacists, and they get used to it, and they realize at the end of the day that they're just as good as the other ones. So it takes time. There's a lot of education to be made, but I'm very confident this can be made because it has been done already with other biosimilars.
I might just jump in that as well, because it shares exactly what I've heard from other type 1 diabetics that I've spoken with. And I think the other pain point in the market to note is the insulin pumps. A lot of them are tied down to a specific insulin brand's pharmacokinetics. So if you need to use a specific brand's long-acting combined with a specific brand's short-acting based upon the brand of pump that you've bought. And so I think that if we're looking at the full top to bottom market, you need to also be thinking about finding a non-delineated insulin pump brand that can work with any kind of insulin and develop the technology alongside the actual biomedicine itself.
Thanks, Alex. Ilya, any thoughts maybe on the second question around how do we ensure that these innovations change the market rather than reinforce the dominance that we see?
It's probably a little bit too early for us to establish territory of a commercial battle with the big three for sure. But our strategy in general has been direct contact with the health boards of specific nations, and our commercial goal is to engage in national distribution through healthcare systems. So when that happens, we're not the one who actually fights for the brand. It's availability of the product that is the most life-saving process. So that's kind of really our hope. If a national effort is amplified, and then we are just delivering affordable insulin. That's kind of our view on it.
Right. We'll take two more questions from the audience, and then we'll hand over to our other experts. Is the first question is, how do you approach getting capital for innovations that are trying to disrupt a very big, very established market, in addition to getting capital for a population that is often overlooked by investors? And then the second question is at what point in development do you engage with the national regulator, or is it still too early? Thank you. So one question about sort of the investment and the capital needed to get these innovations to market, and then the second one more around regulatory aspects.
I can take a second one again, David, if you wish. I think my recommendation, and I have a lot of experience of dealing with regulatory agencies, and as it is from all the speakers today, quite innovative, disruptive technology, the best recommendation is to start a dialogue with regulators ASAP. As soon as you have a couple of analytical data showing similarity or not with the parent molecule, you should engage dialogue and see are they convinced? Does it need more? Is there any concern about immunogenicity? Whatever. So the bottom line is go and speak to your regulators ASAP.
I can answer in a similar way. We're already in contact with the regulators, but again, we're looking at the pathways through individual nations, not necessarily through FDA, and that's one way we're trying to approach it. The challenge, of course, would be if this would be labeled as biosimilar or maybe less than biosimilar. So there is a different ways to look at it. It all depends on how open those national boards are. We're finding it's actually quite surprisingly engaging, but it could be too early to say. You know, some of these markets are. I don't want to say anything negative, but challenging in terms of understanding how politics of local healthcare system works. And in terms of funding, we have 100% commercial, so we survive on or exist rather on the private fundings, and some of the funding comes from non-dilutive funding grants as well, but we're mostly funded by venture capital and family offices. We're trying to diversify targets and not only working on insulin, but on a few other strategies, and that seems to be quite a so far successful. But funding is always for R&D, not only in healthcare, but any type of deep tech is challenging. You know, so it's not only what we're facing; it's any other deep tech company would face.
Thank you for those answers, and thank you participants for your questions. I think we'll move on now to get the perspectives and insights from our three experts. And Julien, thank you for being with us today, and I'll hand over to you to give your thoughts and perspectives on the three presentations and the three innovations that Ilya, Xavier, and Alex have presented.
Thank you, David. Can you hear me well?
Perfectly well.
Okay, thank you. So as a disclaimer, I'm not an expert in insulin at all. I work on neglected tropical diseases inside the MSF, Médecins Sans Frontières, Doctors Without Borders, and I developed some interest over the years on a different class of medicines, which are therapeutic antibodies, including recombinant antibodies. So I will not speak much about insulin, of course, in this presentation because it's not my area, but I will try to extrapolate a little bit what's been said to another topic that was mentioned in several presentations. The case of access to monoclonal antibodies or recombinant antibodies. Because I think there are similar problems between the two categories of medicines. There are differences, of course, as well. So as you probably know, there are many monoclonal antibodies already approved for autoimmune diseases, cancer, chronic diseases, and monoclonal antibodies also have a great potential for infectious diseases that is not really exploited, tapped into very well. There are a large number of candidates for many very important infectious diseases like malaria, Ebola, rabies, diphtheria, HIV. I'm missing quite a few, RSV, of course, and dengue, and a few other infectious diseases with a high public health impact. Inside MSF, we have listed the monoclonal antibodies that are of highest interest for us, and that we would wish to see fully approved within 5 to 6 years, 5 to 10 years. It's already quite a long list, but there are going to be obstacles, and that's what I want to share with you. The first obstacle, I think, is that usually the prices for monoclonal antibodies are extremely high. And the general public, they think that the high prices of biotherapeutics like monoclonal antibodies is due to the high manufacturing costs. But I'd like to caution that vision. The manufacturing costs for monoclonal antibodies are generally much higher, of course, than for small molecule drugs. But this is not the main reason why prices are exorbitant. Prices are exorbitant because there is no robust competition to bring down prices. If you look, for example, at the manufacturing costs, standard manufacturing costs for conventional methods to produce at large scale monoclonal antibodies, we're talking about something like $100 per gram of antibody produced. But if you look at the prices of the different products in high income markets, it's much more like $1,000, and sometimes even more than that, per gram of a monoclonal antibody produced. So the main factor for the high prices currently is the exorbitant returns on investment or profits that are made by the developers, and manufacturing costs are not the main reason. That doesn't mean that we shouldn't look at manufacturing costs and alternative methods to bring them down. I think there's still a case for that, but we need to understand a bit the needs and the market dynamics of the different products that we're looking at. So, for example, if we're looking at a product with a very low dosage, like I'll take the examples of the new RSV prophylactic antibody that is being launched now by Sanofi and AstraZeneca. It's called nirsevimab. It will be launched this winter. The dose is between 50 milligram and 100 milligram. So even if you have a manufacturing cost of $100 per gram, that's still quite affordable. You know, it's between $5 and $10. So is there really a need in that case to invest into different methods that could like halve or reduce by 50% or 75% the manufacturing cost? At the end of the day, the final price will be, the final cost of goods will be a little bit lower, but that's not the main problem. The main problem is that Sanofi and AstraZeneca in the US, they have the intention to charge nearly $500 for that product. So the issue is not the manufacturing cost. If you look at another product, Ebola, one of the two approved monoclonal antibodies for Ebola is a cocktail of 3 antibodies developed by Regeneron. The dosage is extremely high. It's extremely high because I think the affinity of the antibodies themselves are not so good, by the way. But anyway, it's very high, and it's 12 grams of antibodies are needed for an adult weighing 80 kg. So even if you have a manufacturing cost of $100 per gram, that means that the manufacturing cost for your treatment course is already $1,200. So in that specific case, even if the company didn't want to make exorbitant profits, if the company wanted to adopt some sort of a no profit, no loss approach, even with that good intention in mind, it would still be relatively unaffordable. In that case, I think there's a strong case for alternative methods. So what I mean by that is that you need to have a look at the dynamics of every single product, at the capacity to pay, at the cost effectiveness of the different products, at the market dynamics and the needs and the demand, in order to select among the very long list of monoclonal antibodies those that would benefit most from these alternative low-cost manufacturing methods. And the second point that I wanted to make was about conservatism of regulatory agencies. I'm not sure it's the right word, or cautiousness, I would say, of regulatory agencies. My experience, my limited experience, is that regulatory agencies in low and middle income countries are even more conservative than in high income countries. They don't want to take risks, right? So very often they will wait for some approvals in high-income countries and stringently regulated countries before making similar decisions. That's a very basic understanding. Some countries really want to challenge, of course, the regulation on biosimilars and things like that. But for new methods like the ones that you propose, although some products are already in the market, it was mentioned by our colleagues from biosourcing, but these products are not very well known, and they're not used in these countries. So I expect some extreme consciousness or conservatism. So I'd like to hear your vision about that, how we can address that. So my two important messages, as far as I can tell, is it's important to know the market dynamics, the needs, the demand, the size of your market before making the choices to develop that product or this product. Oh, well, it's of course, you know that you belong to industry, so that I think that's a basic part of what industry does, but it's important to reiterate anyway. And my second point is get prepared for regulatory cautiousness. And maybe finally, I think that for products that I'm talking about, like monoclonal antibodies for infectious diseases with a high burden in resource-limited settings, I think we need to also to support kind of public-private partnerships in a not-for-profit with a not-for-profit approach in order to bring these products forward. Because I think we can help as MSF, we can help potentially for clinical studies at some point. Other institutions can help with different types of support. So I think it's individually small biotech companies, it will be challenging to reach the market, but as part of a coalition or a consortium, that may be a little bit easier. So that's basically my points. Thank you.
Thank you so much, Julien, for those points and sharing that valuable insight and experience. Molly, you have a vast experience looking at issues around access to insulin from the different perspectives that I presented in my introduction, from research and development all the way down to use. Interested to hear your insights and thoughts based on our colleagues' presentations. Thanks, Molly.
Hi, thanks again for the presentations. It's super interesting. I mean, I have to say a little, some of that is over my head, but I find it really interesting thinking about manufacturing. And I do, have some of the same concerns, I think, that Julien had and a few of the people have. But I think the new manufacturing, in a way, can be quite exciting. I mean, the idea of thinking about a better thermostable insulin is fantastic. I think that's a great idea. I think some of about manufacturing, really thinking about some of the new developments that can also come with it, in a way, can be worth it in that way. I do, and I think it's already been echoed in some of the comments, it's understanding how from manufacturing we get to a lower cost, high quality product get it in the hands of the people who need it. And I feel like, to me, there's a huge gap between those two things still. I mean, I think, Julien talked about the regulatory agencies and the difficulty there, I think. And I think one of the comments was about people living with diabetes. I mean, I think it's, the example was in the US, which is just switching one analog to another. And I think, in low middle income countries, you hardly see biosimilar insulins. And I think it's more complicated than a lot of other biosimilars because it's such a personal product. And, a lot of the doctors are getting paid by the pharmaceuticals to push a certain product. There's a lot of, biosimilar companies we've talked to, the marketing alone makes it not worth it to get involved in these countries, because it's so hard to get entry into something that, they've only been using one product for years and years and years. So I think it's a huge uphill battle to consider how to show that it's a quality assured product, and that people are willing to not necessarily switch, but willing to, let it into their lives in a way. Then I think, and just one thing, sorry, this just as a comment, Alex, I know you said something about insulin pumps. Insulin pumps can use any fast-acting insulins, and they don't use long-acting. So it's any fast-acting. There's no brand. With with the pens, yes, but not with insulin pumps.
My mistake. Thank you so
No,
much.
it's okay. And then, yeah, I think, I mean, I think just going back to that, with the manufacturer, I mean, I think there is something, as a person with diabetes, thinking about, it was interesting, I didn't know that insulin had been used on made from a plant before, but thinking about, using something that might be in a different way, putting that into your body or thinking about an open source product. I mean, I think it's quite scary just from a personal perspective. And I think there's a lot you're going to have to do to sort of overcome that idea of something different, particularly something that, keeps us alive. So that's just something. Then I think Xavier had mentioned about the environmental, the considerations of environmental sustainability within the manufacturing. To me, I think that's a super critical and important point when considering the manufacturing element. I mean, diabetes is a very wasteful disease. Unfortunately, there's a lot of products that come into play. There's a lot of plastics. And I think we've all been asking for something that maybe could be manufactured in a new way. And considering the environment within that, I think is very important. Then I guess, my two main questions is really how far are we from actually looking at when is this going to, make an actual ripple effect into improving access to insulin for people in need? I mean, where are we in terms of the timeline of this? And how do some of the new things that have happened within the insulin market with reductions of prices in the US, the companies, the big three, sort of trying to make more of an impact in low and middle income countries and actually reducing prices, and how do we fit within that timeline to actually be able to provide something that's low cost and lower than what's there and competitively low? And then also just a curious question about for Alex about the open source insulin and what are the risks in terms of providing open source technology? I mean, I think in technology, at least for CGMs and different other parts of diabetes, it's really changed things for people to be able to have access to free technology that will link them better to their diabetes management. But I think in terms of for medicines, what are the risks there? And how do you sort of navigate those risks so that, you're making sure that people are using these codes or whatever for, something that's going to be safe and quality assured. So that's it. I mean, I think, super interesting and fantastic to hear all of these new ideas. I think I remain a little bit skeptical on how, this is actually going to help the patients have access to safe, quality assured, and most importantly, affordable insulin. So thank you very much.
Thanks so much, Molly, for that feedback and those insights. Amanda, your thoughts on what we've heard, this morning, this afternoon, this evening.
Thank you, David. I want to start with congratulations to the innovators. As Xavier says, this is a quote unquote well-characterized protein. So the degree of innovation to bring it into plants, into milk, and Alex, your efforts towards open sourcing are very impressive. Insulin, unlike other treatments, many other treatments, is a life-saving treatment. It's 101 years old this year. People who need insulin but don't get it die. For other people who might use insulin, for example, people with type 2 diabetes, some 20% in the United Kingdom who do use insulin helps them achieve better control, which in turn helps them to improve their quality of life and potentially increase their length of life as well. So the source of insulin has changed over the years. We heard about animal insulins today, albeit in milk, but we've had animal insulins for years. It required us, of course, to go to the abattoir, the slaughterhouse, to get the pancreases, which is where insulin is made, or pancreata, of animals. But and this is an effective source of insulin, but it's not a sustainable one. And some people have trouble with animal insulins. They actually develop antibodies to them, and others find them objectionable on a number of reasons. You have to kill the animal, and other people object on religious grounds to animal-sourced drugs. So we've also heard about the transition in 1982 to making insulin in bacteria and yeast. And today we heard about a sort of third wave of providing insulin to people. But to do this, these new sources of insulin have to be acceptable to four main groups, in my opinion. The first and most important are the people with diabetes themselves. So the insulins have to be accessible. They have to be easy to store. And I was fascinated to learn that the innovation of potentially making a single chain would obviate or at least improve the storage issues related to current insulins. And for people who must pay for their own medicines, it has to be affordable. I've also learned in my reading about plant insulins that there may be potentially oral sources of insulin made from, as it happens, I think, old lettuce leaves. So if that were the case, that might be an innovation for people who do not choose to inject. Secondly, these insulins have to be acceptable to the regulator. Julian, I believe, said, get ready for regulatory cautiousness. But of course, we do want our regulators to be cautious because they ask important questions. The questions they ask are simple and three. They would want to know if these new insulins are effective. They want to know if these new insulins are safe. And they want to know that these new insulins are quality controlled. But as I heard Xavier say, this is not science fiction, and this is a well-characterized protein. So whether these are biosimilars or a new class will be an interesting question. A third group for whom these new insulins will have to be appropriate, for those of us lucky enough to live in countries with universal health care, is the payer. And the payer's questions will differ from the regulator's questions. The payer won't simply ask, are these drugs effective? The payer will ask, how effective are these new insulins compared to what people would otherwise use, which in this case is a fairly simple answer, and that is insulins made by currently conventional means. If these insulins are deemed biosimilar, they're very likely only to have to be priced at the same or lower price than current insulins. And then it's sort of a no-brainer that they would be the preferred source of insulin. But remember, value for money is different than affordable to a country, and both will have to be met. So these new insulins will have to be deemed worthy of, and they will be, they are life-saving drugs, but the budget impact will also be taken into account. And with 90% of people with diabetes having type 2 diabetes, but 20% of them being on insulin, that's actually the biggest group. Lastly, the clinicians, given that these drugs are not over the counter, will have to be convinced that these are reasonable alternatives because they prescribe them. I'll close then with a quotation from a paper by Bashan called Cell Factories for Insulin Production, where he and his colleagues write, quote, based on the yield, brackets, 3 milligrams of proinsulin per gram of leaves, close brackets, it was estimated that 1 acre of tobacco plantation could yield up to 20 million daily doses of insulin per year. So I think that is a fantastic outcome, more insulin and less tobacco. So thank you very much.
Thank you, Amanda. for that insight, those comments, and sharing those views. Alex, I'm going to put you on the spot to just very quickly react to some of those challenging insights that Julien, Molly, and Amanda have provided us.
Fantastic, my pleasure. So I suppose quickly just responding to Julian and Molly, that I wholeheartedly agree that a lot of the issue that we're facing is actually high profits. And I used to joke back when we were starting off that the first thing we need to do is institute a big price war and try and help drive the cost of manufacturing methods down, potentially just by bringing in more standard manufacturers. Because you look at any other off-patent medication that's chemically synthesized, like aspirin, it's incredibly cheap. And so to me, the answer really comes down to, can we lower the barrier to entry by building a new framework for biosimilar approval? And as Julian was saying, that really starts with the FDA and the EMA, because if you can get biosimilar approval through those groups, a lot of other countries will follow through. Certainly the TGA here in Australia will. Now, I'll just finish off by sort of suggesting, this is a wild idea that's been bouncing around in my head for a long time, is that we need some kind of public-private partnership, not-for-profit company. What that company needs to do is provide an open source repository of biologic vectors for generic manufacture of biomedicines. So effectively, we get a biosimilar approved open source available to anyone bacteria with a DNA sequence in it, everything characterized from how it's from the actual bacteria itself to the purification, a standardized protocol for building a manufacturing plant, and we provide this to any manufacturer willing to enter the market and provide medicines, and have this not-for-profit both just, here's the bacteria with the DNA inside of it, and also here's how you set up a GMP approved plant. That way we can try and bring in more people, and we don't have 100 different companies all trying to go through biosimilar approval so that they can have their own insulin. And because effectively all of that regulatory cost gets added to the cost of the final product. And I just also wanted to quickly, I think a lot of Amanda's questions were for the other two, but I was thinking about a quote I'd heard a while ago about how many pancreases were required before 1978. There was some 63 million pig pancreases per year. And I think a large reason why we no longer eat sweetmeats as a species is because prior to all of the pancreases being required for insulin, they actually were a commonly used meat as well. But anyway, when you take them all to the abattoirs and use them for medicine, you no longer eat them. Anyway, I'll let the other two respond. Thank you.
Thank you, Alex. Xavier, any thoughts on your end from the insights from Amanda, Julien, and Molly? Xavier, we can't hear you.
Right. Can you hear me now? Yeah. So I think we can characterize with sophisticated orthogonal analytical methods the insulin protein very well. Hence, we should not need more than one phase 1 trial to get approval. So we have seen that with pegfilgrastim, where the regulators in US and Europe and Canada, not Australia, I have to say, have accepted pegfilgrastim based on one single phase 1 trial. And I strongly believe, now we have to see, that the same pattern could be applied for insulin. So we get demonstration of similarity from an analytical perspective, and then we run a phase 1 study, and boom, we can find.
Xavier, tell us a little bit more about how that study was actually done. So did people, the same people in a crossover design get the one insulin, then the other insulin, and then doctors are hanging around to rescue them should they need that?
Yeah, and there is actually, I have identified in Germany a phase 1 facility that is specialized in clamp studies. And they probably where we're going to run the study. There's a shop that's been built up by diabetologists, and they are highly skilled in demonstration of similarity of different insulins. So this is probably where I'm going to go for demonstration of biosimilarity, clinical biosimilarity.
I see. So famously in biosimilar things, they have to be equivalent or non-inferior. Nobody knows, well, answering the question, what do you mean by non-inferior?
No, it is not, no, it cannot be non-inferior. It has to be equivalent.
Yeah.
And then the question is the margin of equivalence, which is always a debate. There are two sections. There is the statistical equivalence, and there is the question of clinically meaningful equivalence. And that opens a big can of worms, because what does that really mean? Nobody knows.
Thank you.
But this is an issue we have with cancer drugs development. You know, what is the mortality rate we feel acceptable? Is it 5% difference, 10% difference? It's always a debate we have. It's a complex one.
Mm-hmm. Thank you for those remarks, Xavier. Ilya, on your end, any comments, responses to the challenging points that were raised by our colleagues?
Yes, difficult questions. Very difficult questions. First of all, commenting on plants, it's actually kind of a plus for us, because we by just chance end up talking to quite a few groups in Muslim world, and the idea of doing implants, which would be halal, is kind of came up a couple of times. And actually bacterial systems are not considered to be halal, but there's no other choices. So, you know, that's kind of plays well into what we trying to do. In terms of timelines, you know, we at the moment looking only at GMP certification as our next commercial benchmark. So basically trying to model productions at scale, at a reasonably large scale. In terms of regulatory approvals, we sort of put it a little bit off at the moment, because that's it's not a biggest challenge for us. Yeah, and then I think final sort of comment in terms of price. Again, we are looking, our whole world is cost per gram. So we're looking at actually APIs, and our comparison analysis is with APIs that generic manufacturers are, it's all known, is export prices for Lispro specifically, because we're working on it right now. And I don't want to kind of publicly say it, but it's about at least 1/10 of the bulk price API at the moment. That's why our investors are very excited about it, because that could be a real game changer. So once you kind of reach that level of cost savings and optimization at the large scale, then there is a potential that other barriers might be actually slightly less Bastilian in some ways.
Great, thank you, Ilya. We're ahead of schedule, which is rare. Any further questions from? I see Amanda has already jumped in, so go ahead, Amanda. And for our participants online, please feel free to use the Q&A tab and ask a question or raise a comment. Go ahead, Amanda, please. Your mic, Amanda. Sorry, I don't know if you started.
I thought I'd share the experience of One Insulin as it went through NICE, which is the National Institute of Health and Care Excellence, which makes reimbursement not so much decisions as suggestions to the National Health Service on what to fund. And this is all in the public domain. But when long-acting insulin analogs hit the market, they came to NICE, and the question in the setting of type 2 diabetes was, again, how effective are they compared to what people would currently use, which was NPH insulin, human intermediate acting insulin, and do they reflect value for money? And the newer insulins were fractionally better insofar as there was less hypoglycemia, which could reasonably be modeled out into a better quality of life, although better quality of life wasn't shown in any trials. And yet they were approximately 4 times more. Now, these cost effectiveness analyses will take into account not simply the price of the new insulin, but all of the downstream complications that might ensue, such that if one were associated with a lower rate of hypoglycemia, NICE would take that into account. But despite taking those things into account, it was not deemed good value for money. And so our national recommendations were for type 2 diabetes, if you were starting someone on a long-acting insulin, the recommendation was to choose a human insulin. So one can envision that these, if this new product is quite different, it will go through a same sort of route. But there's also a possibility that if it were even slightly, if you will, less effective, but just a tiny bit less effective and considerably less expensive, as Ilya said, what if it came in at 1/10 the price? The government would also recommend it. So technically worse product, but considerably cheaper because of the savings it would free up. Now, it's very challenging, I think Molly would agree, that if you took an insulin like this to a person in clinic and said, listen, we're going to swap your insulin, and the person would say, oh, is it as good as what I'm on? You might have to say, well, it's almost as good. Which doesn't go down too well in a one-to-one setting, but on a public health population level, it frees up monies to do other things like buy COVID vaccines and cancer drugs and hospices. And so that is just an example of, if you will, a real life experience of a country with universal healthcare system.
Thank you for sharing that experience. Any other thoughts, comments from any of the panelists? Xavier?
Do you think that would go through the MHRA if it is inferior or even slightly inferior?
Okay, so Xavier brings up a good point. At least in the United Kingdom, NICE cannot address any drugs that don't have a marketing authorization.
There you go.
And so, yeah, probably, possibly not.
Mm-hmm.
Possibly not.
Yeah.
So, where these kinds of situations have been seen that didn't involve a marketing authorization, for example, is treating, as it was, hepatitis C for 6 months instead of 12 months. And it was just a tiny bit less effective, but pretty much half the cost. But it was just a different regime, wasn't it? It wasn't a new drug. That's a very good point, Xavier.
Thanks. Molly, please go ahead.
Just 2 points. One, I mean, just on insulin, I mean, it's a really tricky slope. I mean, I think we're now where glargine is on the EML, and I think it's been a long discussion. I understand. I mean, David and I have this argument quite a bit in ACCISS. Clinically, of course, you can say it's not any different, but I think quality of life for people with diabetes, it's a big change for them. And so it's hard to argue. I understand a medicine you take once a day, but it's hard to argue when your day changes every day. If you get 20% less of up and down, then it's worth it. It's hard to quantitatively capture that, but it's a tricky one. And I think if you said, okay, an insulin is 10% less effective, you have to take in mind then the doctors have to change the way that they treat the patients. They have to work with them to readjust their dosages. It becomes a little bit more complicated than that. It's a tricky one, and I totally understand from a public health perspective one way, but I think it's a little, diabetes is a little bit of a minefield in terms of that. And I did just have one other question about I think Amanda actually mentioned that, just about like any thought to delivery devices, or maybe creating an insulin that might be used in a different way that might be more easy for the user.
since it's kind of in the plant fields, yep, a lot of literature around plant-made insulin and lettuce directly consumable. Problem number one, how do you regulate the dosage? So that's really, it's you can't really put the patch on the on the letter. Secondly, they have incredibly low yields. So it's academic research. It's anecdotal, but it can be done. We looked at it, but it's I don't think it's commercially practical. We would be the first one suggesting that it's probably too challenging. So maybe sometimes in the future, maybe Alex is going to come up with bacteria that lives in biome and just secretes insulin when it's needed.
I will admit we did examine that at one point, but and there has been an iGEM team in 2019 that also ran with that idea. But I do think that it is incredibly hard it has to be able to switch it off and not just cause the death of the patient, which it would if you had unrestrained production of insulin. But maybe if you got two species of bacteria working side by side, one making glucagon and one making insulin. Yeah.
Well, that'll be a topic for a next challenge or discussion. Any final thoughts from our six experts today? If not, then sorry, someone was. If not, I just really want to thank Ilya, Xavier, Alex, Julien, Molly, and Amanda for their insights and sharing their experience. And sorry, Alex.
Sorry, we do have a question from Evan and Laura in the Q&A tab.
Sorry.
If you wanted to quickly get that one.
Sorry, I missed those. Apologies. Sorry, thanks for raising that. So we have a question. Hello, wouldn't this suggestion by Prof. Adler that a 10% less effective insulin might be acceptable overlook the issue of additional volume of injection that would be required and thus increase discomfort? And the second question is, I'm also curious about the injection device question from Molly. So I think Molly's question was, are I guess mainly focused on Xavier and Ilya. Are you also looking at delivery devices? Sorry about that. Thanks, Alex, for highlighting those questions.
Okay, I think the first one is, to me, the vast majority of technologies, and by technology, I don't mean a device on your app. I mean, most technologies are drugs in terms of HTA, health technology assessment. Most are better but more expensive. I don't think I suggested that 10% less would be acceptable, but let's look at an example, possibly ad absurdum. But imagine that a new insulin were a billionth less effective because you had one fewer mild hypoglycemia in the remainder of your life than you would have on your current insulin. And let's, again, taking this forward ad absurdum, suggesting that this insulin cost a billionth, the new insulin of what we have now, that almost everybody would choose to spend taxpayers' healthcare money on that. So if it were slightly less effective, the answer to Evan is no. All the dosages and injection problems, everything is taken into account, as is possible in making these decisions. And it is all about opportunity cost, such that these Molly, the quality of life gain is by no means not appreciated. It's just the knowledge that if that money were to be spent elsewhere in the healthcare system, it would likely to bring in much greater health gains. So it's not whether these difficult decisions have to be made, it's how to make them. So and David, after the second question, I actually have a question for some of the panelists.
Okay, yeah, so the next question was about delivery devices. So new ways of looking at delivering insulin to people needing insulin. Ilya, Xavier, are Biosourcing, Neos, and Bio also looking at those innovations?
I'm not personally, but I do know that the Open Insulin Foundation is also investigating various research into, and I don't want to like get anyone's hopes up here, but like because it is still very, very early days, but in looking into actual treatments for diabetes beyond in terms of especially type 1 restoring pancreatic function. I do recall in 2019 we saw some very at the meeting we saw some very interesting technologies in restoring the functionality of alpha cells with genetic technologies, and I do know that the Open Insulin team is looking to try and invest some of the money that they earn into research labs that are looking into that. So obviously the greatest solution to the insulin issue would be to have an actual treatment for diabetes. I personally would believe we'd probably be still at least 15, 20 years off, but there's certainly some very interesting technologies and some worthwhile projects to look into in that space as well.
Xavier, Ilya, this question on delivery, if you want to.
To be frank, we haven't really started to think about it, and anything that adds to cost is something which is kind of prohibitive. So we are focusing on delivering the insulin at the lowest possible cost, and I would say we figure out how to deliver it later.
Same. We're trying not to add complexity. I wouldn't even say cost, cost of opportunity basically. And our for biosimilar indication, we're looking at as standard as possible delivery with single chain. Well, that's the challenge, because if it's a novel indication, then you would have to basically recalibrate everything, and that's it's a part of a cost, and part of a challenge for sure.
Thank you. Amanda, back to you.
Yeah. In my sort of reading round to in advance of this symposium, I read about sort of growing insulin in plants, but then basically eating the plants. So we haven't historically had oral insulins, although there have been efforts with oral and inhaled, because as a protein, it denatures in our very, very acidic stomachs. Can you sort of grow insulin in old lettuce? And the implication was you eat it and all the sort of the plant structure protected it to the point where it could get absorbed, or is that, as Xavier would say, actually science fiction?
No, it's actually not science fiction, but there were attempts to do oral insulins. I mean, there's still a company that raised billions of dollars, still even in existence. It's a question of bioavailability, basically. So it's how much plants do you need to eat, for one dose. So it basically comes out that you would need to eat 2 kilos of lettuce.
Then you'd lose weight, because you wouldn't have any more room in your stomach to eat anything else. So your diabetes would get better if it were type 2 diabetes.
I mean, I think I don't want to open the can of worms, but I think it's kind of, in my view, it's a little bit challenging, if you sort of look at where the market goes with GLP-1 products, it's sort of start being marketed as a sort of substitution of insulin in some bizarre form. I think we need to address current situation. People just need insulin, and large quantity needs to be produced, and I think that's the challenge number one, in the world. I totally agree that analogs have a very, very, perhaps not that much of a market advantage or patient health advantage, but they are accepted. So we also have to take that in consideration. So once they accept it, it's we need to provide it at lower cost and better system of getting into the patients. That's our view. Try to simplify things that are already working and take it and run with it.
Well, it's a drug with a very narrow therapeutic margin, so you have to be able to predict how much insulin there will be in the blood. And after eating lettuce, as suggested, I'm not sure we can predict exactly the level of insulin you're going to get.
It's, you know, I mean, there are other plant hosts that also can work. You can do it in strawberries. You know, we looked at that as well, but you know, again, we're trying to use a source that is well known. So it's bentaminiana is actually nothing to do with tobacco, and then the whole recombinant process takes the any side of nicotine completely out of the picture. But that by itself is a challenge in its own, because people hear tobacco and they think that you're going to be like producing nicotine. So it's even that word is challenging, you know, sometimes with highly educated people. So everything's has to be simplified to deliver the product to people who need it.
Well, great. Thank you so much again, Ilya, Xavier, Alex, Julien, Molly, and Amanda for your insights. I think to piggyback on what Ilya has just said is I think today highlights that we are facing with a challenge. As has been mentioned, insulin was discovered 100 years ago, over 100 years ago, first used 100 years ago, yet we're still facing this challenge that many people in need cannot still access this life-saving medicine. And I think it's exciting to hear these three innovative approaches and this change of paradigm that was mentioned, and how this change of paradigm also has impacts beyond just how we make insulin, but as Molly mentioned, is the environmental impact of how insulin could be made using goat's milk, using plants, and also a change in paradigm that goes beyond just insulin. There's also this mention of public-private partnerships. Is that also a change in paradigm in terms of how we see medicines being made, and not just being in the public-private domain, but a shared domain between public and private entities and non-governmental entities? And finally, does this change in paradigm change the market that we are all familiar with, that unfortunately is not working and not solving a global health problem? I think what was also exciting in hearing these different approaches is the adaptability and scalability of these models, and also the opportunity for local production of insulin versus these large capital-intensive production methods. But I think as was highlighted, beyond the manufacturing, the downstream elements and the gaps that exist in getting insulin to individuals within health systems, the issues of availability and affordability and the price of insulin remain challenges. But it's exciting to see how potentially changing one piece in this complex chain could fundamentally change what we do and how we see the insulin market. But I think as Amanda nicely highlighted, we still need to address, the vision of these different partners and stakeholders, whether it's regulators, payers, prescribers, people living with diabetes, with regards to these innovations, and what do they mean in terms of are people ready to take insulin that's made in goat's milk, in plants, or other ways. But that said, I think, as the need is there in terms of ensuring available, quality assured, affordable insulin to many millions of people globally, I think this is an interesting way, and we look forward to hearing how this moves forward. And I think having innovation and open science as part of solving the solution sort of brings us back over 100 years ago to innovators, Professors Banting and Best at the University of Toronto, who did things differently, and were able to fundamentally change the course of type 1 diabetes and diabetes in general with their discovery of insulin. And also that Professor Banting is often said as saying that insulin belonged to the world and not to him. And can we, through these innovations and through these innovative approaches, bring that sort of mantra back? So again, a huge thank you to Ilya, Xavier, Alex, Julien, Molly, and Amanda for sharing their insights, and to all our participants. We will make the video of this event available as soon as possible. And again, thank you again, and everyone have a great weekend. And we hope to be organizing other events like this, and of course, we'll share those with you as they get planned. So thank you again, and à bientôt, as they say in French.